What if I told you that the biggest lie about autism and brain change isn't that our brains cannot change, but rather how that change actually happens? Here's what the marketers won't tell you. Your brain absolutely can and does rewire itself throughout life. Environmental factors genuinely influence how your genes express themselves. These are not myths. They are documented scientific realities. But here's the deception. Companies have taken these legitimate discoveries and twisted them into promises that you can fundamentally transform autistic neurology into neurotypical neurology. With headlines like these, brain training that eliminates social challenges, or genetic optimization that unlocks normal development. And this one, supplements that rewire sensory processing. The lie isn't about whether change happens. The lie is about what kind of change is possible, beneficial, and ethical to pursue. Today, we're going to understand what neuroplasticity and epigenetics actually do in autistic brains, examine the evidence for what works and what doesn't, and explore what the most powerful interventions support autistic neurology rather than trying to eliminate it. Because the real science reveals something revolutionary. When we stop trying to make autistic brains neurotypical and start supporting them to function optimally as autistic brains, remarkable adaptations become possible. This episode isn't just an academic exercise for me. It's personal. Many years ago, I was in the depth of the worst autistic burnout of my life. My mind, which I had always trusted, felt like a shattered map. I desperately searched for help anywhere I could find it, and in that desperation, I became the perfect customer. I bought the supplements that claimed to regulate dopamine and serotonin naturally. I even purchased an expensive meditation program which claimed to help rewire my brain to find its center again. I devoured books on neuroplasticity because I was desperate to understand what was happening to my mind, which none of the psychiatrists and therapists who were treating me could identify beyond deep depression. The search for answers in the conventional medical world led to a dead end. In fact, it led to one of the most chilling suggestions I have ever received. We need to give you electroconvulsive therapy. Yes, ECT. The idea was to zap me out of a depression they could not define, a diagnosis they were never able to identify, and a burnout they didn't have the language to name. That was the moment the mission for this work began to crystallize. I realized that the territory of brain science, our very neurology, was a place where hope and exploitation lived side by side. I had to learn to navigate it myself, not just to survive, but to understand, to look for answers where I couldn't find any. Welcome to NeuroRebel, where we cut through the noise to explore what science actually tells us about different minds. I'm Anita, your host and fellow navigator of neurodiversity research. Today, we're examining territory where legitimate neuroscience meets predatory marketing, where hope collides with exploitation, and where understanding the difference can save you from costly mistakes while revealing genuine possibilities for growth. This episode matters because the autistic community faces a unique form of scientific exploitation. Real research on brain plasticity and gene expression gets repackaged into interventions that promise to normalize rather than support autistic development. If this conversation challenges assumptions you've held or helps you navigate these complex waters, please share it with someone who needs this analysis. Your support helps other families find evidence-based information. So let's start with precise definitions, because clarity protects us from those who profit from confusion. Let's start with neuroplasticity. It is the brain's capacity to reorganize its structure and function in response to experience. Let me break this down to what it actually means at the cellular level. Think of your brain as containing billions of tiny communication points called synapses. Imagine them like bridges between brain cells. When you practice a skill repeatedly, those specific bridges get stronger and more efficient, sort of like a footpath that becomes easier to walk as more people use it. This is what we mean by strengthening synaptic connections through repeated use. The brain also prunes unused pathways. It's literally like eliminating connections that aren't being used, sort of like a gardener removing dead branches so the tree can put energy into healthy growth. Meanwhile, in certain regions, like the hippocampus, the brain can actually generate new neurons throughout life, adding fresh cells to existing networks. And finally, let's talk about functional networks. Think of these as neighborhoods of brain regions that work together for specific tasks, like language or attention, and they can reorganize their communication patterns based on experience. Let's move on to epigenetics. It involves changes in gene expression. And here I need to clarify what gene expression actually means. Your genes are like a vast cookbook containing thousands of recipes. Gene expression is the process of actually making those recipes, like deciding which ones to prepare, when to prepare them, and how much of them to make. Environmental factors can influence these decisions without changing the recipes themselves. So stress, nutrition, and social experiences act like signals telling your cellular kitchen which genetic recipes to prioritize and which ones to put away. Some of these changes can even be passed down to children, like inherited cooking preferences that influence which family recipe gets made most often. Both processes are continuous and measurable. Brain imaging studies document structural changes from interventions. Epigenetic markers can be tracked in blood samples. The question isn't whether these mechanisms exist. They absolutely do. The question is what they can and cannot accomplish and how to harness them ethically and effectively. Here's what the evidence shows they cannot do. Transform fundamental neurological architecture. An autistic brain developing better emotional regulation skills through mindfulness practice, for example, remains authentically autistic while functioning more effectively. So the neuroplastic changes support autistic neurology rather than replacing it. Now let's ground this in a metaphor that captures the complexity without the oversimplification. Think of neuroplasticity like the way established forests adapt over time. When certain pathways through the forest get heavy use, they become clearer and more efficient to travel through. When pathways fall into disuse, on the other hand, vegetation reclaims them. But the forest also develops new routes when environmental conditions change, like after storms or seasonal shifts or changes in wildlife patterns. Crucially, these adaptations happen within the forest's existing ecosystem. A deciduous forest developing new pathways remains deciduous. The fundamental character persists while specific routes and connections evolve. Now, for autistic brains, research suggests that this adaptive process may work differently than in neurotypical brains. So studies using transcranial magnetic stimulation, for example, show that autistic adults exhibit what researchers call hyperplasticity. This heightened plasticity creates both opportunities and vulnerabilities. It may contribute to intense pattern recognition abilities and detailed processing strengths, but it can also lead to sensory overwhelm and difficulty filtering irrelevant stimuli. That is, the system is highly responsive, sometimes too responsive. And understanding this helps explain why environmental modifications can be so powerful for autistic people. Small changes in sensory input, routine structure, or social demands can create disproportionately large impacts on functioning, both positive and negative. The key insight is this: autistic brains aren't less plastic than neurotypical brains. If anything, they may be more plastic in certain ways. But this plasticity operates within autistic neural architecture, supporting better autistic functioning rather than seeking a neurotypical transformation. Let's turn to environmental signals and genetic expression. Epigenetics helps us understand how environmental factors influence autistic development without changing core neurology. Research shows autistic individuals exhibit significantly higher rates of what's called epigenetic drift. That is the gradual divergence of gene expression patterns over time, compared to neurotypical control groups. This enhanced drift appears to originate during early fetal development when neuronal replication peaks. So think of it like trees responding to environmental signals while maintaining their species identity. Prenatal exposure to maternal immune activation, certain medications, or nutritional factors can influence gene expression patterns that affect neurodevelopment. But these influences modify how autistic neurology develops and not whether it develops at all. Studies in animal models show that prenatal valproic acid exposure can induce autism-like behaviors and create epigenetic changes that persist for at least 3 generations. This demonstrates genuine gene-environment interaction, but the resulting neurology is consistently autistic in character. So what is the practical implication? That environmental factors matter enormously for autistic development, but they influence the expression of autistic traits rather than the presence or absence of autistic genes. Optimal environments support clearer communication, better sensory regulation, and enhanced executive functioning within autistic neurological parameters. So this research validates the importance of environmental accommodation while rejecting the fantasy that the right environment can eliminate autistic characteristics entirely. So now that we understand how neural change actually works, let's examine what the research shows about popular interventions claiming to harness these processes. And let's begin with mindfulness-based interventions, which have gained considerable attention in recent years for supporting autistic adults. Mindfulness involves training attention to focus on present moment experience without judgment. It's like learning to observe your thoughts and sensations as they arise without immediately reacting to them. Systematic reviews show that this approach significantly reduces anxiety and depression in autistic adults, with moderate effect sizes around 0.5. Neuroimaging studies document genuine structural changes. They show increased cortical thickness in regions crucial for interoception and emotional regulation, and enhanced connectivity between executive control networks. A pilot fMRI study found mindfulness increased functional connectivity between the cingulate cortex and the sensory motor regions during self-reflection in autistic adults, and correlating this with reduced depression. Now, this is a measurable neuroplastic change supporting better autistic functioning. However, traditional mindfulness approaches often ignore sensory sensitivities and communication differences. Adapted approaches show better outcomes when they account for autistic processing styles. Now let's turn our attention to Cognitive Enhancement Therapy, or CET for short, where we encounter one of the more rigorously studied approaches. CET combines computer-based training in cognitive skills like attention and problem-solving with group-based social cognitive coaching. Think of it as mental gymnastics paired with real-world practice sessions. Randomized controlled trials show this approach improves neurocognition, social cognitive skills, and employment outcomes in verbal autistic adults. Now, this represents a genuine skill building through neuroplastic mechanisms, but it happens within the autistic cognitive architecture rather than replacing it. This intervention works by providing structured, long-term practice in attention and problem solving combined with social cognitive coaching. And success rates for competitive employment increase significantly compared to control groups with this approach. But when we turn to neurofeedback, the evidence becomes more philosophically interesting, let's say, and definitely problematic. And what is neurofeedback? It involves monitoring your brain waves in real time and providing immediate feedback. So imagine watching your own brain activity on a screen and learning to consciously influence those patterns. The premise of this holds a certain technological poetry, the idea that we might observe our own neural rhythms and learn to conduct our inner orchestra with greater intention. Yet, this appealing concept runs into empirical reality. While some studies report benefits, systematic reviews consistently find inconsistent results, poor study quality, and effects that often are indistinguishable from placebo. The theoretical mechanism, that is, training people to consciously control brainwave patterns, really lacks strong empirical support. Lived experiences vary dramatically, with some autistic adults reporting temporary benefits that fade over time, while others are finding the process overwhelming or stressful. And let's move on to commercial brain training programs, where we encounter perhaps the most pervasive example of how legitimate research gets distorted for profit.
Say, make money, money, make money, money, money.
These types of programs promise cognitive enhancement through repetitive exercises. For example, digital drills designed to sharpen memory, attention, and processing speed. Now, the marketing is interesting because it appeals to our desire for self-improvement and suggesting that we can level up our minds like characters on a video game. But the evidence tells a more sobering story. While people improve at the specific trained tasks, meta-analyses show minimal transfer to real-world cognitive abilities or daily functioning. The companies cite neuroplasticity research to promote these programs, but the plasticity involved appears to be narrow skill acquisition rather than broad cognitive enhancement. And this creates a crucial distinction. neuroplasticity that builds specific context-dependent skills versus plasticity that enhances general functioning. Finally, let's consider repetitive transcranial magnetic stimulation, or RTMS, an approach that represents the cutting edge of neuroplasticity research applied to autism. RTMS uses magnetic fields to directly stimulate specific brain regions, offering unprecedented precision in targeting neural circuits. Emerging research suggests protocols designed to reduce excessive long-term potentiation may help with sensory sensitivities and executive function difficulties by modulating the hyperplasticity we discussed earlier. Now, early studies show promise, but this area remains investigational, and this approach targets the specific neurobiological mechanism rather than attempting broad behavioral change. The pattern that is emerging is this: the interventions with solid evidence that support autistic neurology functioning better as itself, and those with weak evidence typically promising to make autistic brains function more like neurotypical brains. So, understanding legitimate neuroplasticity research protects you from predatory marketing that twists these concepts for profit. For example, direct-to-consumer epigenetic testing represents a particularly egregious example of this. Companies offer to analyze DNA samples and claim to identify, quote, root causes of autism, and then they sell you personalized supplement regimens promising to, quote, optimize gene expression. And the problems here are fundamental. There is currently no validated epigenetic test that can diagnose autism or reliably guide treatment. The supplements marketed based on these tests lack evidence for the benefits they claim. The entire model exploits parental hope while misrepresenting entirely the genetic complexity of the human brain. Now, this differs entirely from legitimate clinical genetic testing. For example, chromosomal microarray or specific condition screening, which has diagnostic yields of 10 to 30% and identifies genuine medical needs requiring management. So companies selling intensive behavioral interventions increasingly use neuroplasticity language to justify approaches that remain focused on normalization rather than support, because they claim their programs, quote, rewire autistic brains toward neurotypical functioning. The research shows that these approaches can change behavior through what's called repetitive conditioning, but this comes at a significant psychological cost. Studies have shown and documented increased anxiety, depression, and post-traumatic stress symptoms in autistic adults who experienced intensive early behavioral interventions. The neuroplastic changes achieved often involve suppressing autistic characteristics rather than building adaptive skills, and therefore creating what researchers call shallow compensation, which is a fragile behavioral change that breaks down under stress. Now, let's examine the supplement industry's exploitation of epigenetic research. Marketing claims about supplements that, quote, support healthy gene expression or optimize neural development typically extrapolate far beyond available evidence. While nutrition genuinely affects neurodevelopment, the leap from folate influences methylation to this specific supplement formula will improve autism symptoms simply lacks scientific support. The most harmful aspect isn't financial waste. though families often spend thousands of dollars annually in these industries. But the harm is in the underlying message that autistic neurology requires fixing rather than supporting. So, what does evidence-based support for autistic neuroplasticity actually look like? Well, let's start with environmental optimization, which sometimes produces the most dramatic improvements. Sometimes the most powerful interventions involve environmental modifications that allow natural abilities to emerge, like adjusting lighting, for example, reducing auditory overwhelm, or creating predictable routines. These changes can dramatically improve functioning by working with autistic sensory processing rather than against it. Research shows that environmental accommodations often produce larger improvements in quality of life than behavioral interventions attempting to change the person. And when we turn to skill building, the most effective approaches work within autistic frameworks rather than against them or trying to eliminate them or change them. Effective interventions teach specific skills while respecting autistic communication styles, sensory needs, and processing differences. This might include, for example, social skills training that validates autistic interaction preferences while building workplace navigation abilities. The key principle is this: building capacity rather than eliminating characteristics. Now let's turn to addressing co-occurring conditions, which represents another crucial area where neuroplasticity research offers genuine benefits. Autistic people experience anxiety, depression, and other co-occurring conditions at higher rates than the general population. Evidence-based treatments for these co-occurring conditions, that is, those adapted for autistic processing styles, often produce significant improvements in overall functioning. Finally, let's consider the importance of supporting natural compensation rather than forcing specific behavioral changes. Effective support helps autistic people develop their own adaptive strategies. Research shows autistic individuals often create sophisticated compensation mechanisms when given appropriate scaffolding and environmental support. And here, the goal shifts from normalization to optimization, and that is helping each person's autistic neurology function as effectively as possible, rather than changing it. So here's the fundamental tension we're navigating. Neuroplasticity and epigenetics are real, powerful processes that offer genuine opportunity for growth and adaptation. But the question isn't whether change is possible. It is what kind of change serves autistic people's authentic development versus what serves other people's comfort or wallets with autistic differences. The research reveals something crucial. When we stop trying to make autistic brains neurotypical and start supporting them to function optimally as autistic brains, remarkable adaptations become possible. Mindfulness can reduce anxiety while preserving intense focus abilities. Environmental modifications can eliminate sensory overwhelm while maintaining enhanced pattern recognition. And social skills training can build workplace navigation while honoring authentic communication preferences.
Yes!
Now, all of this represents a paradigm shift from deficit-based interventions toward strength-based support. So instead of asking, how do we fix this brain? Let's ask, how do we create conditions where this brain can thrive as its own autistic self? The implications extend beyond individual interventions to research priorities, educational approaches, and social policy. When we understand autistic neurology as different rather than disordered, we invest in accommodation rather than normalization, and understanding rather than elimination. The most exciting frontier isn't technologies that promise to make autistic people neurotypical. It is in research that helps us understand how to support autistic neurology reaching its full potential. The science is clear. Your autistic brain can and does change throughout life. Environmental factors influence gene expression. Neural networks reorganize based on experience. These aren't marketing claims. They are documented realities. But understanding these processes properly protects you from exploitation while revealing genuine possibilities for growth. The question isn't whether neuroplasticity exists in autism, it's how to harness it ethically and effectively. When you are evaluating interventions, ask yourself this: does this approach support my autistic neurology functioning better? Or does it promise to change me and make me neurotypical? Does it build on my natural strengths and processing style? Or does it try to eliminate my autistic characteristics? Is the evidence behind it solid? Or does it rely on testimonials and theoretical extrapolations? Let's do an exercise. This week, notice when you encounter claims about brain optimization or genetic reprogramming, and consider whether these approaches honor neurodivergent development or exploit hopes for normalization. You've been listening to Neuro Rebel. I'm Anita, committed to cutting through pseudoscience to find genuine insights about neurodivergent minds. If this analysis helped you navigate these complex waters or evaluate interventions more critically, please share it. You can find resources and transcripts at neurorebelpodcast.com and follow us on social media at NeuroRebelPodcast. If this work adds value to your life, consider supporting it. Not because you owe us anything, but because together we can keep building understanding for all the travelers navigating these territories. Buy me a cup of coffee on my website. Remember, your autistic brain isn't broken machinery requiring repair. It's a complex system capable of remarkable adaptation and growth while remaining authentically, powerfully itself. Until next time, thank you so much for listening. Keep questioning, keep growing, and keep being beautifully, unapologetically yourself.